behalf of all, my sincere apologies for the technical glitch that had happened for a few minutes. So we are again resuming, and now we have the presentation by Mogili Ramaiah from Entomology, presentation number 8. Okay, Mogili, go ahead. 1 minute. I have already lost.
There is a quote by Charles Elton: "The progress in taxonomy will indicate the progress in other branch of sciences." A very good evening to all the dignitaries, respected teachers, and my dear friends. I welcome you all to my presentation. I am Ramaiah from Division of Entomology, and before going to the introduction, so I would like to explain briefly about the bamboos. As we know, the bamboo is the integral part of our livelihood. So during the World War II, the bamboo Taj Mahal dome was covered with the bamboo scaffolding. So there are many publications, articles which are related to showing the significance and importance of the bamboo. So what are these bamboos? So bamboo is a special group of plants from the beginning of human civilization. If you see in India, India has blessed with more than 148 species under 29 genera, occupying 60 million hectares and standing the second largest country after China. And if you see in India, the central part and northeastern part is the major. So applications of bamboo, there are many, and majorly paper and pulp industry. And these bamboos are threatened by many biotic and abiotic stresses. So within that, the insect pest of bamboo is the major. So starting from the defoliators to up to root feeders. So if you see the diversity of bamboo insect pest, so here the more than 600 species of the Hemiptera is there. So within the Hemiptera, there is a tiny beautiful creatures created by the god called as leafhoppers. So what are these leafhoppers? So as we know, insects are the most abundant organisms on the earth. And within the insects, there is the Hemiptera having 11%. And within the Hemiptera, there is a largest superfamily called as Membranoidea. And Membranoidea is having the 6 families. And within that, Cicadella is one of the largest family, having more than 30,000 species. So why it is important? What is the need to study these leafhoppers in relation to the bamboo? So as we know, our agricultural food plants as well as bamboo belong to the Poaceae family. There is a chance of force shifting in the future because of the global climate change as well as the extensive cultivation. And the Legion in 1919 reported that there is a specific tribe called as Mucarini, which is exclusively feeding on the bamboo. And also here Mucalus splendida, which is belong to the Mucarini tribe, which is vector for the phytoplasma. So in relation to the leafhoppers, with respect to bamboo, so what is the research gap from India? So there is no systematic assessment of the leafhopper from India and limited taxonomic studies. Only line diagrams are available. There is no colorful for easy identification, and the diversity is unexplored. No data available with respect to the molecular characterization. To overcome this lacuna, we frame the title that is Biosystematic Studies on the Leafhopper Species associated with the bamboo from India, with the objectives of exploration, morphological characterization, identification of the leafhopper species associated with the bamboo, and to prepare the methodic checklist and diagnostic key to the genera and species of bamboo leafhopper, and to study the phylogeny of the leafhopper species associated with the bamboo. So if you see the first objective, a diversity has conducted to explore the bamboo diversity from India. There are 35 locations of 12 states we covered. So these leafhopper samples we have collected with help of the sweep net, aspirator and light trap, and finally the processing was done by established procedure and photographs taken with the stereosome auto montage. And after that, male and female genitalia we dissected, and that male and genitalia parts we transferred to the genital capsule for preservation. So what morphological characteristics we used for the taxonomic studies? So these are the habitats. So there are whatever bird type space. So like that, the pronotum, face, male genitalia, female genitalia, we will describe. And in the case of when we are describing the new genus, so we will see the legs and wing characters. So in the case of the leafhopper, male genitalia is very important for the species description. So which is having subgental plate, connective, vocal, styly, eriagal dorsal and eriagal lateral. So species description as follows, and finally holotypes, paratypes were designated and deposited in the National Pusa collections. So this is the overview of the leafhopper fauna associated with the bamboo from my work, that we describe redescribe the 43 species under 30 genera, having the 5 subfamilies and 12 tribes. So within that, as I told, Mukarine, which is exclusively feeding on bamboo, which is the major pest. So here the Mukarine showing the highest number of the percentage of the pest. So these are some of the illustrations of the bamboo leafhopper we described, excluding the new species, new taxa. So these are some of the female leafhopper illustrations. So new taxa we discovered from my study, that is the 4 new genera, 10 new species, and 2 genera which are new to India. So if you see the 4 new genera, that is bamboo paga from Andhra Pradesh, Niranjana from Assam, Shanaya from Uttarakhand, and 1 genova we didn't keep the name, so it is from Meghalaya. So these are the 2 genera, that is Bambusi mucaria and Mahonia. Previously we reported from the China, and previously it is reported from China. Now from my work it is reported. And this is the overview of the 10 leafhopper new species from my study. Along with the leafhopper study, I also collected the hemiptera diversity during my study. Second objective is to prepare the annotated checklist diagnostic key to the genera and species of bamboo feeding leafhopper. A total of 91 species and 35 species. So we prepared the checklist by isolated publications and zoological records, CABI, and the different leafhopper websites as well as the Google Scholar. So from these we collected compiled the data and make into a checklist. So the checklist including the subfamily, tribe, genus, species, synonym, type locality, type deposited, and distribution. So diagnostic key prepared for easy identification, for rapid identification. So here from the subfamily level to up to species level, we develop a diagnostic key for the easy identification of the leafhopper. So third objective is to study the phylogeny of the leafhopper associated with the bamboo. For authentic identification, as well as if you want to see the evolutionary relationship, so it should be morphotaxonomy integrated with the molecular taxonomy. In molecular phylogeny, the DNA extraction PCR amplification is the first step. So here DNA was extracted from the leg or whole specimen, and DNA is amplified by PCR with the different primers, and sequencing we sent for the outsourcing, and we submitted to the NCBI. These are the different primers used during the study, and these are the more than 60 sequences submitted to the NCBI. If you see, the phylogenetic analysis was conducted with available sequences as well as the own sequences. So here the histone 28S sequence were aligned with the help of the muzzle application in Mega software, and unlike genes were integrated into a one data set using the Mega software, and finally the maximum likelihood analysis was done. So this phylogenetic analysis suggested that whatever the most of the new genera we described, so it is according to the morphological characters. So here during my study, the collections from the different states. So I also observed the maximum color pattern diversity in the population of the Mucaria splendida. So here if you see, there are 10 color marks from each sex. So each color mark we described as a type 1, type 2, and so on. Likewise the females also we described, and when we dissected the genitalia, so the genitalia was same. So these morphological characters we integrated with the molecular taxa, so finally confirming that morphologically all specimens of Mucaria splendida differ in their habitat coloration, but whereas genitalia and genetic composition of all the specimens irrespective of the color is similar. Similarly, we have also reported a distinct morphological variant in the Mucaria Viraktamath, which is recently published in the new taxa. This type of integrative approaches helps in resolving the complexity identification. So because to avoid misidentification, misinterpretation by the immature taxonomist, so this type of integrative approaches are helpful. So from my study, I also reported bamboo as the alternate for the mango hopper. So in a nutshell, a total of 43 species, out of which 10 were the new species which I discovered from my study. So with this, I would like to mention my silent research findings. So we recorded a total of 53 species of leafhopper associated with the bamboo from India, and discovered the 4 new genera and 10 new species, and 2 genera which are new to India. And I told the Mucaria tribe which is exclusively feeding on bamboo. So for that tribe from my study, I added 26% which are new to science, and provided the complete annotated checklist as well as the diagnostic key for rapid identification of the species. and more than 40 sequences first time deposited to the NCBI and molecular phylogeny was worked out and provided integrative approaches to resolve the complexity and identification of the species, and bamboo reported as the alternate host for the mango hopper. So future prospects need to study the vector status of all these leafhoppers, and information on the biology of these leafhoppers is not lacking, and the host association and different bamboo species yet to be studied. And as we know, the invasive pest regularly reporting. So careful monitoring and timely reporting of pest instances is very necessary to avoid any future outbreak. And new species, we registered even to do bank for unique ID. From my work, I have published 3 research papers, 2 in zootaxa having the nerve rating of 7.09 and 1 in insects having a nerve rating of 9.14. I attended the conferences and got the 2 best, received the 2 best posters. One is at international and one is at national level. And I attended the trainings and workshops conducted by the NAHEP and DSP. And I am also an associate member and reviewer for the magazines and journals. And even I elected as a fellow of the prestigious Entomological Society of India. And I attended the webinar conducted by the JSI, NIPHM, NBIR. So with this, I heartfully acknowledge my chairperson and members of advisory committee, and head and professor, my esteemed teachers, Division of Entomology, Director ICAR-IARI, and Dean PG School, and UGC for fellowship, and NPC labmates, seniors, friends, and juniors. Finally, I would like to end my presentation with the quote by father of taxonomy Carl Linnaeus, that is, if you do not know the names of the things, the knowledge of them is lost too. Thank you.
Yeah, Dr. Mr. Ramaiah. Just 2 questions mainly. You have used 60 taxa.
Total 43, sir.
43.
Checklist prepared for 91.
How many sections do they know?
Like 5 subfamilies, sir. And 2 types, sir.
How complete is the extent of sampling in this subfamily? How complete is the sampling process for the variants within a subfamily? What I mean is, all the species
No, sir.
taxonomy strata or hierarchy. Did you sample all the variants? See, you are talking about why I am asking you because you are talking about phylogeny reconstruction, right? The authenticity of the phylogeny that you consider depends on how much complete the sample is.
Yeah. Actually, during my collection, I didn't get like because of the resources as well as the COVID period, I didn't went for the large number of collections, sir. So whatever I got, like new species, I got only 4 or 3. So from that, if I do the molecular work, we have to deposit the holotypes in the museum, sir. So like that, so I tried my best level to take the DNA from the leg, sir. So how extend it came, and I ran, sir. And I faced the problem.
Did you use any sequences deposited already in CBI? For the COI sequences for your target group, there might have been some sequences
already. When I searched, no, sir. So Mediana constricta and there are studies also doing, sir.
Okay,
but that was pretty.
It's okay. The next question is, you have used only mega for analysis. Why not more specific phylogeny reconstruction softwares like ND, COP, that type of program, which give you better flexibility in positioning the taxa?
I will continue my work.
Yeah.
Recently, I have been selected for ICAR.
And also, there is a software known as Mesquite that helps you in evaluating the phylogeny or the clades and positioning. That also later on.
I will do.
Okay. And then you are not part of See, unless you have that internal collaboration, the work is not going to succeed.
Thank you, sir.
Yeah, thank you, Mogili.
Yes, sir.
Good luck.